Thank you. Very nice presentation. I have actually two questions. The first one would be, do you know anything about D2 expression in high-fat diet? You mentioned that there could be ectopic D2 expression in the liver under certain conditions. And let's say if there would be D2 expressed when you induce high-fat diet, that could be an alternative explanation for why you see a phenotype in the knockout.
So the first question as to whether there's D2 expressed in normal genetic situations on a high-fat diet, yes. I don't think there is, not to my knowledge. Only in that LXR knockout mouse. Okay, the second question, sorry.
The second question I haven't asked me.
Okay.
So the other one was, did you check, I mean, this when you check on the genome-wide scale for DNA methylation, you will end up with lots and lots of hits, which most of them might be false positives. Did you check using pyrosequencing as well, whether the sites that you really overlap with your gene expression would be differentially methylated if you take larger groups of animals?
I do not believe they did that. They just tried to kind of use the Venn diagrams to look for any, commonalities between the transcriptional changes and the methylome changes.
Peter de Lange, Italy. I would like to ask you, you showed the expression of genes involved in lipogenesis in embryonic animals versus adult animals. And in the end, when you showed the methylation-based assay, you came up again with strep BP, which is a very important lipogenic gene. And in the adult animals, then you showed, before you did the methylation analysis, that those genes were not elevated.
Correct.
Did you measure also thyroid hormone levels in the embryonic and in the adult animals? And how do you envisage, because T3 binds to positive TREs, and all these genes have positive TREs in their promoters, and how do you envisage that during development this simple way of activation of gene expression does not occur anymore?
Right. So I'm not sure if they collected, if they measured T3 levels from the embryologic animals, but I believe they did from P1, and I showed some, that it was normal and the same. And also that within the liver was the same. So I think that I don't fully know, but I think it's just pretty remarkable that a very transient peak in T3 is so important for controlling, really, there's an epigenetic role of that surge in T3 that persists for lifetime and affects downstream, long-term gene expression profiles.
Domenico,
Okay.
and then...
Hi, Elizabeth. So thank you for this nice presentation. So do you know where and when the albumin is expressed in the liver? Because one of your hypotheses is that other cells than hepatocytes are expressing D2. So where exactly are you depleting D2?
So the albumin is not really expressed until the late teens of the E. So it starts around E15-ish, the albumin. So only then would this be affected. And what's really important around P1 is that there's major changes in the liver around that day and induction of hepatocytes, because the liver in embryologic mice doesn't have that many hepatocytes. But then the albumin starts being expressed in the last few days of embryogenesis, and then the liver really changes into its adult more form around P1. So this is a time where there's major changes in the liver cells. What cells the D2 is in, I don't know otherwise around that time, but there is some D2 expression and activity. As I showed at the beginning, we know it's in macrophages, but the function of the liver is so different for an embryo. I'm sure that there could be some role in those other cells as well. I don't believe so. I believe the albumin, the reason we did it is because it's hepatocyte specific.
Elizabeth, here. Very nice talk.
I have a couple of questions. The first one is somehow related to what Jans made it, and is essentially, do you know whether, for example, if you induce a sort of liver regeneration with hepatectomy, you observe again a peak in type 2 deiodinases? And the second one is more general. Your hypothesis is that you have a sort of developmental defects in your mice due to something going on early postnatally. Do you think that if you provide an extra amount of thyroid hormone in that windows, you can rescue the phenotype that you are observing later on in life?
So the second question first. That's a very interesting question. And I think that that question is difficult to answer because if you were to give T3 on postnatal day 1 systemically, there would be a lot of other effects as well. Dr. Bianco talks about there being a time-sensitive period for T3 in which several different developmental processes are known to be affected even during due to very small, short duration surges in T3, such as puberty is something that can be affected, and the liver, and a few other things as well. So I don't know if you could rescue the phenotype, but it would be difficult without a liver-specific thyroid hormone beta agonist, I guess. Okay, the first question was
on hepatectomy to see whether you have a kind of
so we have not looked at that.
Okay, so and if there is one more question, then we'll have to stop.
Lizzie, I was very interested to see in the liver development, it looked as if type 3 deiodinase expression is paralleling the profile of type 2, if I understood that correctly. So it's a little bit different from many other systems where there's a reciprocal exchange of the deiodinases. Do you have any idea what DIO3 might be doing at the same time as DIO2?
I don't know, but I agree with you that the fluctuations in DIO3 mRNA levels during that period are very interesting, and that definitely deserves more study that I don't think we have done yet. But you're right, it's interesting that it isn't the opposite of D2, as we see in brown adipose tissue.
Don't you think this could be related to different cell types?
Absolutely. Like we said, this is a very exciting time in liver development. A lot is changing at that time, and it absolutely could be due to fluctuations in the proportion of different cell types around that time in development.
Okay, thank you very much for this very nice talk.